Organ-on-chip are small plastic devices that contain real human cells and tissue. This living tissue is perfused with a liquid. It transports nutrients, oxygen, and metabolites, just like blood does in the human body. With a chip, scientists can track oxygen levels, cytokine release, and many other factors. And they can add elements to the mix, like immune cells. That is what the microorganolab in Tübingen has done. When it comes to testing immunotherapies, their latest OOC models hold great potential over animal models because
for everything related to the immune system, the species differences are very, very big. So an animal might have a very different immune response to a certain stimuli than a human. Then also on the other side, most immunotherapies target very, very specifically human targets.
Human targets like cancer. Cancer cells are malignant degenerated cells. They grow unchecked because they have developed ways to hide themselves from the immune system, specifically from its T cells. In CAR T-cell immunotherapy, T cells are taken from a patient. In the lab, they are genetically engineered to recognize cancer cells. These now upgraded CAR T cells are then re-injected into the patient, where they identify, attack, and destroy cancer cells. How well they work varies from patient to patient, and there is significant risk of side effects, which is why it is important to test how human cells react, something the breast cancer on chip allows.
We can actually follow how these CAR T cells migrate into the tumor microenvironment, how they start to attack the tumor, but we can also follow on how they start to release cytokines, which then go back into the perfusion and are then being transported to peripheral organs. And we can look at all of these different key events happening in this chip in live and in a kinetic manner.
It's textbook knowledge reproduced because you always learn it should be like this, but to see it is really fascinating. So, yeah, and I mean, what literature cannot tell you is the thresholds, and there you can clearly see that we can test if a CAR T therapy is working against a certain type of cancer or not.
Testing if and how well a therapy is working for an individual patient helps researchers design treatments that are more effective and safer. A leap in personalized medicine. The new organ-on-chip also helped to understand other important aspects of how the human immune system works.
So what we are able to do with our lymphoid tissue chip is to model an adaptive immune response, for example, the production of an antibody upon the delivery of a specific antigen in the chip and outside of the human body.
For me, it's really interesting now to use it for different drugs and to run tests on different drugs with this system. And also, actually, even the lymphoid tissue on chip is further in development now. People interested to try to modulate infections, or people trying to model autoimmunity, which is also partly happening in lymphoid tissues. And then you can use this model to test treatments again.
And that's another advantage over animal models. Because chips are smaller, easier to control, and can be tested in large numbers at once, they tend to provide results much faster and cheaper. With more accurate data and faster results, the new organ-on-chip models help make immunotherapies better and safer for all of us.